from one amino acid to one alpha keto acid. But we are just switching the amine between two different compounds so that we never produce the free amine at that time. We can produce a free amine, but we don't want to produce it inside the muscle tissue.
You said the enzyme that converts pyruvate to oxaloacetate, it's the same, it can do the same, but reverse? You can do oxaloacetate to pyruvate, is another enzyme?
No, it doesn't go backward. We don't go from oxaloacetate to pyruvate, we never do that.
Oh.
It's only one way. But I say it can be shared between two different processes, either TCA cycle or gluconeogenesis.
Okay.
But we don't go backward. We don't go from oxaloacetate to pyruvate. So you see, we can actually make the cycle to go about 60 to 100 times faster by just increasing the amount of oxaloacetate by amino acids that we have. Or even from glycolysis, if we have so much pyruvate, they can boost the cycle so fast, circuit goes about 60 to 100 times faster. So basically, produce more ATP by just making cycle faster. Now, if you look at the citric acid cycle, one question would have been, is there at any time that we can, okay, see here, cycle is going to citric acid cycle, was there any time that we could have produced sugar from a cyclical way? You see, we want to produce fat. Well, what if we wanted to produce carbohydrate, sugars? We can't do that. So that's, if we consume fat can never turn into sugar into a body. We can't do that. But bacteria can do it, plants can do it. How would a bacterial plant be able to change sugar, fat into sugar, while we cannot do it? Well, they have something else called glyoxysome, and glyoxysome uses something similar to TCA, but it's missing the two enzymes that produce CO2 from a cyclical way. So it means they don't lose the CoA carbons, they actually keep them. So you see how the isoscetate lies on the top, malate synthase in the bottom, which bypasses the two enzymes we have, so they keep the carbons in. And look what they produce, glyoxylate. Now glyoxylate is produced as two carbon compound, and then producing succinate, which is similar to malate, and it moves, these two then move to the mitochondria now, and they become glucogen. So these bacterial plants can consume fat, and they can produce carbohydrate. Now you can see if you have seeds, plant seeds can grow very easily, and they're full of fat, because they can produce no sugar. But we can't. So we cannot do this because we're missing glyoxysome. So the good thing is that since bacteria, they have glyoxysomes, if we're infected with bacteria, we can actually kill the bacteria if we make any drug targeting these two enzymes, we don't have them. So we only have to block isoscetate lyase or malate synthase to go. So for example, tuberculosis has been caused by bacteria that remains on our tissues, consumes the lipid inside the membrane, and is hidden for a long time. You could have, individual could have that disease and they could walk without knowing that they actually have the disease because the bacteria remains there and it consumes the lipid. So basically it not been detected. But then one simple medication that blocks these two enzymes can kill the bacteria because we're missing those two enzymes. So very easy to fight them back if we know what other thing they do in biochemistry part that we don't do. So that does not harm us if we block those enzymes. Not I'm saying that if you block any enzyme it's going to harm you, but if you do not do that, then look what you have to consume. Acid. Because then you can kill the bacteria and you harm yourself. So I'm saying that compared to arsenite, which is not selective, block the TCA, those drugs are very selective because they only go after glyoxysome, not going after mitochondrial function. So that's what it is. So now we can kind of put it together. We have so much glucose, we have done so much glycolysis, we have no need if this is inside the fat tissue, inside liver tissue, we have no need for acetylcholine anymore because every of these tissues, they understand that there's so much ATP because insulin is telling them there's so much ATP, there's so much glucose, nobody needs anything from you. So what do? They're going to change acetylcholine to fat. So if you look at the fat tissue and the liver tissue, they're all producing fat at that time. Now in order for the muscle to be able, and the fat tissue to be able to take as much as glucose they can, the glucose transfer number 4 is being controlled by insulin. So it means they come into the membrane, they're going to take the glucose out of the blood. So we have increased the efficiency of glucose uptake from the blood, so they can go. Now certain enzymes, again, they have to be switched on. This is a bit tough for you to see right now, but pyruvate dehydrogenase is. Acetylcholine carboxylase is involved in fatty acid production, so you haven't heard about that enzyme yet. That has been switched on with insulin too, so we can make fat. Fatty acid synthase has been switched on, so we can make fat. Glycogen synthase has been switched on. And enzymes involved in gluconeogenesis, on the other hand, don't have been either inhibited or the transcription has been lowered. So when insulin shows up, it says we don't need the gluconeogenesis, so we don't need those enzymes, and if they're around, they're going to be inactivated. So fructose 1,6 bisphosphate, which was the other enzyme, the enzymes that work against PFK, it has to be an inhibitor. And we wanted the PFK to be active. So there's sequences of events happening. Glucose into the cell has been increased. All the enzymes involved in glycolysis, the regular PFK is active. Pyruvate dehydrogenase complex is active. Fats are produced because we have fatty acid synthase to be active. And we have acetylcholine carboxylase and cytosol to be active. So every signal is green when insulin comes to production of fat from carbohydrate or production of glycogen from glucose in both the liver, the muscle tissue, and the fat tissue. So we can consume the glucose really quick in a very short period of time. So again, if we look at muscle, increases glucose uptake, it makes glycogen. Fat tissue increases glucose uptake, it's going to make fat. Liver tissue is going to increase it too to make glycogen, increase production of fat. At the same time, it's going to regulate and lower gluconeogenesis. The same thing, the reverse will happen when epinephrine comes. We're going to break down the glycogen in both the muscle and the liver tissue. The glycogen breaks down. Liver tissue is going to do gluconeogenesis to resupply the glucose, and the fat tissue is going to release the fat. So it becomes secondary source of energy, so we don't have to rely so much on the glucose at that time. We have a replacement as source of energy, which is fat. So all these things happens when these two hormones are there to just make sure that we have what we need, which is the ATP. Now, how much ATP do we use? We use our ATP. I showed once before that if you think about it, in every hour, if we were not able to regenerate ATP from ADP, we had to consume 1 kilogram of ATP, 2 pounds of ATP. So if you are not going to survive, and we have no talent for producing original ATP, we have no mitochondria to be able to make ATP, we had to consume 2 kilos, 1 kilo of ATP every hour to just maintain metabolism. So what we're doing this, we're consuming 1 kilogram, but we regenerated ATP all the time. So ATP goes to ADP, ADP goes back to ATP, and we keep doing it the entire time. So it's really, it's a lot of ATP. Most of it, of course, is being consumed by the brain. So it's 20% of the energy consumption. It's being used by potassium sodium pumps, which are mainly inside the membrane and they maintain the potential. So a lot of our pumps, active pumps, is that the membranes are consuming. So you can't just say if I don't move, I don't need 1 kilo of ATP. No, even if you don't move, you still need that 1 kilo of ATP. So it's a lot, but we regenerate ATP the entire time. So we make it and we consume it. I was saying before that another thing we have that is a complication we have is that our glucose inside the blood can never drop below 5 liters. So we always maintain the glucose concentration at about 5 liters. At no time it can go lower. We do go higher than this, but then we lower it. But we don't go below 5 liters. Anything below 5 5 millimolar can cause brain damage because of those active transporters are not going to be so active. If we go below 2 millimolar, then we will be in coma. Then we will die. So it shows you again, wrap it up together, it shows you how all this has been coordinated when insulin comes. Insulin is affecting the fat tissue, so they can take more glucose, make it to fat. Insulin affecting the liver to do the same thing, producing fat and glycogen. So it can be shipped, that fat is being shipped to fat tissue. So the liver is helping the fat tissue to have more fat. So they ship to fat tissue. And the center muscle is producing more glycogen to making sure it has reserve when the glucose is not there. And when the glucose is low inside the blood, then epinephrine glucagon will show up. They do reverse. The fat tissue releases the fat. That's fatty acid. We're going to learn that in that chapter, little chapter. Another chapter actually, a couple more chapters from that. Fatty acid will release from the fat tissue. Glycogen is turned into glucose to use for glycolysis. Glycogen goes to glucose to be shared in the liver tissue. Gluconeogenesis is activated so that glucose is produced from amino acids that can be shared. Well, so those are really the main thing we do on an everyday basis to make sure that we have enough glucose into the blood and we have energy sources, which are mainly the fat tissues. We have so much of it. So again, it shows you how this signaling pathways work together, which is shown here. But let me very quick before I finish this. I'll go back to the chapter that I did not cover. Cover the I showed it to you right now. That what was it that I just told you today that we had on that chapter. What I forgot to show you was this, that so let's show you today right now that when oxaloacetate wants to go out of the mitochondria, it goes in form of malate. So malate has that NADH. So malate goes inside the cytosol. It goes back to oxaloacetate, but malate, the hard guardian is on the cytosol. NADH is produced, and NADH can be used for gluconeogenesis. That's something I forgot to show you that we had last time. It can also go as aspartic acid. Then it goes as aspartic acid. It can just be the oxaloacetate, but it produces no NADH at that time. Phosphoenolpyruvate can move between the mitochondria and between the cytosol. So the times that phosphoenolpyruvate can go to the mitochondria, our mitochondria has phosphoenolpyruvate carboxykinase. So, what you get out of phosphoenolpyruvate carboxykinase? Phosphoenolpyruvate carboxykinase enzyme, what does it give you?
CO2, carbon dioxide.
The enzyme phosphoenolpyruvate carboxykinase changes oxaloacetate to PEP, and PEP goes out. Right? Xander will get oxaloacetate out of mitochondria. Oxaloacetate is going out of mitochondria as a form of malate. It can go as a form of aspartic acid. But it's going to go as a form of phosphoenolpyruvate because PEP carboxykinase can change oxaloacetate into phosphoenolpyruvate, and it can go out and it can go to the So it's showing you that gluconeogenesis can get its carbon source out from oxaloacetate in three forms. Malate can go out, aspartic acid can go out, or it can go out as PEP, and it can be used the way it is, as a PEP, with PEP carboxykinase enzyme that changes oxaloacetate to phosphoenolpyruvate. From phosphoenolpyruvate, it directly goes to gluconeogenesis because it's right after pyruvate. So we're actually producing what we need for gluconeogenesis. So I'm showing you that there are different ways of getting out